And I've tried to make this talk as, I guess, as simple as I could, not necessarily providing you lots of complex biology, but more trying to describe to you what we know, or at least what we think we know about biology, and how this could potentially translate into good things for the patients with regards to therapy. So I will cover rapidly mechanisms of resistance to ADT, so CRPC mechanisms, then mechanisms of resistance to the new drugs with very recently emerging data, abiraterone and enzalutamide, and just a word about mechanisms of resistance to taxanes. So just for you to remember how it works, and I'm sure most of you are very familiar with that. The testes are making most of the circulating androgens, testosterone, but the adrenal can also make other androgens that potentially can target the AR. And until recently, we didn't have much treatments to target that. We now have abiraterone and some experimental drugs on top of that. Now, an important discovery of the last 10 years was the evidence of a kind of autocrine secretion. So in other words, cancer cells have the enzymatic machinery to make their own androgens, their own fuel, or at least to convert weaker androgens made by the adrenals into more potent androgens. Now, of course, the androgens will enter the cells, bind to the androgen receptor. There is dimerization here. And as you all know, the ligand NAR complex becomes able to enter the nucleus and bind to the DNA. Again, we have a number of potential inhibitors of the AR. Bicalutamide and enzalutamide are on the market, but also a bunch of other drugs. All right. So, mechanism of resistance. This is a quite old figure. I think it's 15-year-old. So we know something for some time now. Two, I would say, main mechanisms that were identified a long time ago. One is for the cell just to make more copies of the androgen receptor so that there is a higher likelihood for just one molecule of testosterone or DHT or whatever androgen to bind to the androgen receptor. Okay, so by either amplification or other mechanisms. A second mechanism that was identified a long time ago is that of mutations. For a long time, we didn't really know whether that was truly a true human problem, if you will, because it was mostly identified in the lab and with evidence that some cells with AR mutation can be stimulated not by androgens, but not anymore by androgen, but by corticosteroids or even drugs such as flutamide, bicalutamide, etc., etc. But whether that was also true in human was more debated because the mutations are actually rare in localized disease. So those were two mechanisms, identified a long time ago. Then other mechanisms included stimulation by other pathways. The AKT pathway was identified actually a quite long time ago. Again, we didn't really know whether it was truly relevant for patients. And then non-AR related pathways, BCL2, P53, the neuroendocrine cells, which are here. So again, most of this is known for probably 20 years. Now, the consequences for the patients were really minimal, to be honest. The big thing is that the androgen receptor is still the main player. This is, for example, an experiment we did again a long time ago. This is a human prostate cancer that grows in animals. All these brown spots are cells positive for the AR. After castration, it goes down, but when cancer grows again, while on castration, you see that the AR is there. And if you look at downstream genes, it's really up. So the pathway is up. The cancer cells love the androgen receptor pathway, even when we are arming them with castration. And if you don't believe mice, you can look at humans. These are more recent humans data. And you will see that in primary and untreated prostate cancer, DHT in tissues is higher than testosterone, while in a metastatic castration-resistant situation, it's the opposite. It's more testosterone that you will detect in prostate cancer cells. And again, this is quite important because it's not in the blood. We're all measuring, hopefully, all measuring testosterone in a castration-resistant prostate cancer, but the blood is not telling us all the truth. What's happening in the metastasis is different as compared to what you're measuring in the blood, unfortunately. So it's getting more complex. But still, you see that the androgens are there, and that's mostly testosterone in metastasis. So this is a more complex figure, but just to show that based on the simple or simplistic figure I just showed you, there are now many more mechanisms of resistance that have been identified. I won't go into too much detail again, but just for you to understand the big things. Again, one big thing is increase androgen receptor expression, amplification, other mechanisms. That is quite frequent. Mutations, and I will come back to that. I already alluded to that. Now, two important mechanisms of resistance that were identified more recently include AR splice variants, and I will explain what that is, and also increased testosterone production by cancer cells. Again, 10 years ago, we didn't know that. All right, so those are two major mechanisms of resistance. And the rest was plus or minus no. Again, co-regulators, etc., alternative signaling, etc., etc. Can we use this knowledge to help all the patients with new drugs? Probably yes. And actually, regarding the androgen making, we've been using abiraterone, and probably this is part of the success of abiraterone. Abiraterone is really preventing the making of androgens, not only from the adrenals, but also in cancer cells. So very likely, this is why abiraterone works. Some other compounds have failed their development. HSP inhibitors, same thing. Now, one important thing, and I will come back to that, is whether we can invent new compounds that can target the end-term part of the androgen. And this is, for example, the AP01 or AP, or all the AP drugs that are currently under development. And you can also circumvent all this by using a taxane that works a different way. Now, one of the big issues that we still see, not in all patients, but in some patients, is the neuroendocrine differentiation. Because this is a really nasty thing, to be honest. And sometimes it happens like you see a patient who's doing well for a while, benefiting for various androgen receptor targeting, castration, enzalutamide, abiraterone, whatever you want, and then bing, it's exploding, mostly in the liver, and that's a different disease with p53 abnormalities, etc., etc. So it's a different beast. And to be honest, we don't know how to best treat it. Every time I'm being asked the question how best to treat it, I have to recognize I don't know. We can try this or that, but it doesn't work very well, to be honest. So still remain to be invented. But please remember, this is not in all patients. Even when you look at all patients dying from their disease, they didn't develop neuroendocrine differentiation for all of them. Now, can the genomics help us with regards to the mechanisms of resistance? I think Noel already alluded to that on Friday, but it's really a beautiful paper. I really recommend you to reading this one. And this is a big effort, international effort, to try to identify what are the key players for cancer cells to become resistant to all the drugs we have. Obviously, the AR pathway is still key. It's in the center of a game. So if ever we're able to invent new compounds more potent against the AR pathway, we'll win. Now, besides the AR pathway, the PI3 kinase is there, and I will explain briefly what that is, and DNA repair are emerging rapidly as potential target for treatment as well. So this is, for example, the Olaparib data that we've got from the Rose Modern group, and it's really showing to you two things. A, targeting DNA repair defect work in some patients very nicely, nice response with quite, let's say, quite non-toxic drugs. And B, you can predict who's going to benefit based on BRCA2, ATM abnormalities and some other genes. So really beautiful work. And based on that, there is now a lot of PARP inhibitors under study. Second example, targeting the Akt and AR pathways. So those are, if you just look at me, those are two pathways. One is the AR, from the membrane to the DNA. The second one is the PI3 kinase Akt pathway, and they're parallel. So every time you're arming one with castration, abiraterone, whatever you want, the other one will be up, and vice versa. If you're arming this pathway, the AR will be up. And this is what is being shown right here with all these arrows that you see. Okay? So, again, very beautiful work in the lab. And if ever this is true, then you just need to arm the two, to target the two pathways to perhaps induce a better outcome for the patients. We just had the first data indicating that this is likely true. This just came out a week ago at the ESMO meeting, and that's a randomized trial testing abiraterone with or without a drug that is targeting the Akt-PI3 kinase pathway. And as you will see here on the slide, in patients with PTEN loss, so the Akt pathway is on, it seems to work. The combo does better than abiraterone alone, whatever the dose. Now, in cancers with non-PTEN loss, no effect at all. All right? So again, in one hand, you have a drug that perhaps works, and on the other hand, you're able to predict who is going to benefit. So we're really going toward personalized medicine. Very recent data. Of course, we need confirmation, but that was predicted by the lab. What about mechanisms of resistance to abiraterone and enzalutamide? That's emerging as a major problem because we're all using these drugs. They work very nicely, etc., but eventually all patients develop resistance, all of them. And again, this is very recent data I will present to you. Some of them need confirmation, but I think that's really promising. So first of all, AR gains, so more copies of the AR and mutations are associated with primary resistance to these drugs, as you will see here. So for example, you'll see the mutation here in quite light blue. All patients with these mutations resisted to abiraterone. Okay, not a single patient could benefit from this drug. Now, AR gain, more copies. You'll see here that there's an enrichment in patients resisting to abiraterone. The dark blue boxes are patients with AR gains. While patients with AR copy number neutral, in yellow, most of them are benefiting from drug. So it's not, again, not perfect, still not perfect at the moment. It's not black and white. But we're doing a better job, better selecting patients based on that. And of course, we need to better understand what's ongoing. Another paper from another group going the same direction. Very nice figure. I like it. You see, this is progression-free survival. All these patients are, so all these arrows represent a patient. And of course, the longer the arrow, the better the progression-free survival. And you will see here that patients with mutations, these green boxes here, are mostly associated with resistance to enzalutamide. Same for AR gains, the red here, you'll see here. Most of them are resisting, while patients benefiting, and especially benefiting long, don't usually have AR gains. Okay, so it's really emerging as something big. And as you can see, this is something you can detect in the blood. So you don't need to harvest a biopsy from the metastasis. So what about splice variants now? Let me explain that again with my arm, perhaps easier. So this is the androgen receptor. It has a C-term part and an N-term part. The C-term part is the one where testosterone or other androgens will bind. Okay? Then the N-term part is very important for the androgen receptor to enter the nucleus and bind to the DNA. So it's the one that is biologically active, if you will. Now, in some cancers, this part, the C-term part, my hand, is missing. It's not being made anymore. So two consequences. First one, this AR is constitutively active. It doesn't need any more testosterone to go into the nucleus and be active. Second consequence, if I'm using enzalutamide, let's say, bicalutamide, whatever drug you want, it won't find its targets because enzalutamide bind here. All right? So this was predicted, again, in the lab, and it seems to be true. Those are, for example, patients who were treated with abiraterone and enzalutamide, pardon me, and you will see that most patients here in red with AR-V7, so splice variant V7, positive cancers just resist to these drugs, okay? Just because they can't find their target. While all patients, or almost all patients without V7 positive cancers are sensitive. So very promising biomarker, but this is, I will say more about it. So, and taxanes are different beasts. They don't work the same way as compared to abiraterone and enzalutamide. So many patients with AR positive cancers, AR-V7 positive cancers, do benefit from taxanes. So if this is confirmed eventually, it could be nice. You would test the patient. If it's V7 positive, perhaps no abiraterone, no enzalutamide, go straight to taxane. If it's negative, just abiraterone. So this was recently confirmed by another group. I won't go into too much detail, but it's really the same data that you see. V7 negative cancers benefit, V7 resistant, V7 positive cancers resist. Now, very importantly, you need to know that V7 cancers increased with treatment exposure. It's quite rare in a patient who hasn't seen any of his drug. You know, just maybe one or two patients. Now, in the second line, third line, you see the yellow boxes, so V7 cancers. So the more you will give abiraterone and enzalutamide, the higher the incidence of the V7 positive cancers. And again, because cancers are adapting. So, and I will finish with this. What about the mechanism of resistance to taxanes? Because all this biology is really emerging very nicely with abiraterone and enzalutamide, but what about taxane? Unfortunately, I won't have much data to show you because we still don't really understand why a cancer is truly sensitive or resistant to taxane. We have some glimpse that, for example, patients with Gleason 7 cancers may benefit more than does Taxol when used upfront together with castration. This is, for example, a trial we conducted, and you will see visually that patients with Gleason 7 cancers benefited much greater than does Taxol chemo than in differentiated cancers. What are the biological basis for that? I just don't know. Just a fact that we saw. But it was seen across trials, JETUC 12, JETUC 15, Chartered, Stampede, all over the time, we'll see the same trend. Not necessarily significant, etc., but the trend is there in all trials where we're comparing chemo versus ADT alone. So what could be the biological basis? Perhaps ERG. ERG is an oncogene that is, if you will, overexpressed in half of prostate cancers. And we've looked at ERG positive expression, and again, we saw that perhaps patients benefiting from chemo are ERG positive cancers, but again, this is very preliminary. We don't really know whether this is true or not. Same for cancers with RB loss. That's also a very nice hypothesis from the lab, but we need confirmation in humans. So again, for taxane resistance or sensitivity, we will need more data. And that's important. We don't want to expose a patient to chemo for nothing. So in conclusion, better understanding of mechanisms of CRPC in the last 10 years or so, yes, for sure. The AR, the androgen receptor signaling, remains a key element, definitely. With many different means, it's really a key element. PI3 kinase, AKT pathway, and DNA repair response are promising targets from genomics analysis, but also now from a therapeutic standpoint with the PARP inhibitors and with the AKT targeting. Neuroendocrine differentiation remains an issue. Very difficult to know how to best target that. And taxane resistance, I just alluded to that. Now, having said that, if you have a problem and you don't know how to fix it, because we've done our best in the last 20 years to fix the CRPC problem, perhaps we should circumvent the problem. And what could be circumventing the problem? Maybe immunotherapy. If you cannot target cancer cells yourself with a drug or with radiation or with surgery, perhaps we should ask the normal body to do it for ourselves. And I'm presenting this data because it's very recent, just a week ago it was shown. Until recently, we believed that PD-1, PD-L1 targeting didn't work in prostate cancer, based on first analysis we had 5 years ago, but it's not true. Beautiful presentation last week at ESMO showing that some patients benefit tremendously from immunotherapy. Look at this, PSA 70, 46, 2,500, down to undetectable. This is not something you usually see with chemo or abiraterone and enzalutamide, etc. And this is something we saw in the past with ipilimumab, which is an immunotherapy. The issue was that difficult to know who was benefiting or to predict, and a toxic drug. We now have next generation of immunotherapies with PD-1 and PD-L1 inhibitors, and if this is true, and I believe it is true, we now need to identify who benefits. And patients who benefit typically benefit very long. And some patients with ipilimumab are perhaps cured. Second trial, just shown the next day, supporting the same data. So again, immunotherapy work, we might need to better integrate it into all our different strategies. Thank you very much.